Welcome to this Tech Theater. This is about ISO updates to ISO 10993-1 and the new ISO 21726. It's a really exciting time in medical device regulations and biocompatibility. There's been really a lot of things going on, a lot of like big changes, not just like little iterative things, but a lot of stuff going on. And we're going to talk about just a little bit of those today. Before I get too far into that, let me go ahead and give you a little bit of a brief personal introduction. I don't want to spend too much time here, but basically I have an academic background, grew up under a rock, spent all my time reading books and playing with my TI-89 calculator. After spending a few years in academics, I decided to do something else and moved to Nielsen Labs, where I've been there working with them, focusing on chemistry and biocompatibility and how those two areas intersect with each other. One of the things that I really love is helping manufacturers save time and money by testing smart. And to me, testing smart means, really limiting the number of unnecessary tests that need to be undertaken in order to prove that a device is safe. So what I see at Nielsen Labs still is every day we have the same types of materials that have gone through the same processing being tested over and over and over again. It's not really necessary, right? So I want to limit that as much as possible. Everybody wins that way. There's this topic of updates to regulations and biocompatibility and chemistry. This is something that if I had, were allowed, I could stand and talk for, a couple of days with just, stopping to take, a few sips of my Mountain Dew down there. But there's not enough time for that here today. We give seminars that are a lot more in depth than what we have time for here. So if you get a little taste of this and you decide that you want some more, then you can visit the seminars page at Nielsen Labs or just email the seminars@nielsenlabs.com and get whatever information you need. We do a 3-day seminar that focuses on validations of everything, and we do biocompatibility seminars and so on. So feel free to reach out if you want some more. So as I was saying a minute ago on the title slide, things are changing, a lot of things. So just for example, there's the new ISO 10993-1 that came out in August 2018. And then shortly after that, they released another version of 10993-1 that just fixed some minor typographical errors and things like that. There's the new ISO 21726. This came out in February of this year, so that's still pretty fresh. 18562, that's a little bit older, but still very, very important. And not everybody yet is fully aware of what this means and are using this. This is for gas path devices. There's the new ISO 10993-18. This is in FDIR right now. So this completely changes the framework for how we do chemistry testing in support of medical device biocompatibility. So that's in its final form. There's just going through some minor revisions now. And then of course, everybody knows about the MDRs. This deadline, May 2020, is coming up very quickly. There's a lot of talk and chatter about this. I won't spend too much time in this presentation talking about the MDRs because we have another presentation at 2:00 where I'll be co-presenting with Bo Rollins on this topic directly. So there's a lot of things going on, a lot of big things going on. These aren't like minor tweaks to the way that we think about biocompatibility for medical devices. It really is a big shift. So in this presentation, we're just going to be focusing on 10993-1 and the 21726 documents and what this means for you. So I think that the key thing from the new 10993-1, if I had to sum it up in a nutshell, is a really solidified shift from checkbox testing to using a risk-based evaluation type of approach to addressing biocompatibility. And if you go through that document and you start to look for mentions of chemistry testing or non-animal testing ways to evaluate risk. It's all through that document everywhere. So it's just replete through the document. And that's kind of like characterized by how here they have replaced all of these X's with E's, right? So the E's in the table A1, the annex for 10993-1, the E's mean that you should evaluate that endpoint. And we use the word evaluate because we wanted to really emphasize that that doesn't necessarily mean running a test. So you should do something, have a written process, do some sort of evaluation that demonstrates that risk isn't an issue for your device. And that doesn't necessarily mean a test. So that's really like the key conceptual change. Now, how you go through this process to address biological endpoints without testing, there's kind of multiple ways that you can do that. And I'll go through an example here in a minute. There's also a lot of useful information in that annex in the form of these footnotes. And I just took a snapshot of the corner of this document so we can point out a couple. So the footnote G is referring to this column of X's that was in table A1. So if you notice here, that first column, physical and or chemical information, that's an X. And the X means that this information is required as a prerequisite for risk assessment and testing, right? So it means that you need to have chemical and material information for your medical device in principle before you decide to make a test or to do a test. So this is really new for a lot of people. So, maybe you come from, you have a background with a company where you really know what's in your devices, like all of the materials really specifically and their formulations and additives and so on. But you might not. And if you're in one of these situations where you don't know what's in there, then it's time to start to gather that information. So the idea here is before you decide to do an animal test, you need to justify the use of animals or doing that test because of some gap in chemical information. All right. The E's mean endpoints to be evaluated, as I was mentioning before. So I just really want to emphasize this. Having an E in that chart doesn't mean that you need to test something. It means that you need to have some written documentation that risk is evaluated. So this is a really important point. So you notice D here says if the medical device can contain substances known to be carcinogenic, mutagenic, or reproductive toxicants, then you need to address that risk. So here there's pretty good overlap with what we're seeing from the MDRs, right? So part of the MDRs is that you need to understand your materials and know if there are one of the newer requirements in the MDRs. So you need to show that your devices don't contain materials that have a chemical that's known to be a carcinogen, a mutagen, or a reproductive toxicant above a threshold of 0.1% by weight in the material. And it's the same thing here in 10993-1. And then there's this subpart I. It says tissues, includes tissue fluids and subcutaneous spaces. And then for gas path, and it goes on to say that if you have gas path devices, you should use a standard that's specific and relevant to that class of device. So this is really important to understand that if you have a device that fits into some subcategory of devices that has its own special standard, like the dental standard or the gas path standard or the extracorporeal circuit standard, then you really should be using that standard above 10993. So if you have a gas path device that's all dry gas pathway, 10993 says go to 18562. And the testing burden associated with the gas path device in 18562 is much, much less than what you would do if you tried to force it to fit within the framework of 10993. So I think of all the changes in this document, you really should go through and read it carefully yourself if you're in the business of biocompatibility for medical devices. These are what I think are the four most important pieces, and we can pull it right out of the annex table there. The prerequisite to know what's in your materials, the point that you should be evaluating rather than testing. Remember that you need to know about CMRs for your materials, and that wasn't a requirement before. And that you have device class specific standards that you should follow. And that if you do follow them, you can really, really reduce your testing burden a lot. Okay. So as I said, part of this is, using a risk-based approach to evaluate the biocompatibility of your medical devices. So how do you really do that? So the idea is that you should go through some sort of 3-step process where first you take a look at your device and the information that you have on your device, and you identify any gaps that might be there, any like safety gaps or biocompatibility gaps. At that point, then you would make a plan on how you're going to address those gaps. So this so-called biological evaluation plan, not only is it a very, very good idea, but it's now required, right? So in 10993-1, it requires that you have a biological evaluation plan, and if you submit to the FDA and you have this nice, wonderful docket of information for your 510K, and you don't have a biological evaluation plan, then there's a good chance that it will get kicked back. So you must start with a plan that demonstrates that you understand the materials and the processing that goes into your device and what risks those are. And then, once you have the plan and you've established what you're going to do to address these risks, then you go through the process of addressing those risks, either through written evaluations or testing if necessary. Then finally, you have a summary report where you explain to the regulatory body, here's our device, these are the risks that were identified, here's what we did to mitigate those risks, and in conclusion, since everything passed and the evaluations are favorable, the device is safe. So this final report, the biological evaluation report, I found to be really, really important. So there's a lot of packages that cross my desk where the device is great and they mitigated all the risks, everything looks good, but they didn't explain to the FDA why all of this information means that their device is safe. So you got to bridge that final gap at the end for the regulatory body. So in this initial risk identification process, you can use things like the flow charts written in the document to help you identify risks. And I'm not going to go through this in detail, but if you take a look at the document and start to ask yourself some of these questions, like does the device have the same chemical composition as a predicate? If you can say yes to that, then you're already stepping closer and closer to having risks identified without needing to go down this testing path. So we go through this whole process asking ourselves these questions and identifying risks, and all of that comes into this biological evaluation plan. I just want to give kind of an overview of what should be in this type of a document if you're going to go through this yourself to comply to the new version of the standard. So first of all, there should be a complete material characterization of what's in your device. So that includes the suppliers and the way that those materials contact the patients, specification sheets, and so on. Of course, you need to describe the device and how it's used and how it's categorized. If there's any special test sample preparations, that should be in the biological evaluation plan. And then if there's risk assessments or evaluations that you can do right at the outset. So for example, if your device is made out of CP titanium and you know that this material is intrinsically not carcinogenic, then you could say that there and say, okay, look, this is why carcinogenicity isn't a risk for us because of X, Y, and Z. You can address it right in there. Another important part of the biological evaluation plan, and one that I want to kind of give it an example of here, is that this is another good place for you to specify and justify what your family groupings will be. So family groupings are a way for you to take a whole collection of devices that need to be brought into compliance and hopefully identify just a few of those that you can use to test or evaluate, and then extrapolate those conclusions to all the other members of the group. And so one of the biggest, or I guess the most significant, sources of inefficiency in this whole process when I work with manufacturers is that, they're not family grouping. And so they're doing, maybe 10 times the number of evaluations that they really need to do. And then that really extrapolates directly into 10 times the cost. And you can imagine that, some of these tests and evaluations can be, pretty expensive. If you can reduce 90% of that, that's a big deal, right? So I would say that it's worth it to spend time carefully thinking through your family groupings to try to make sure that the results that you have can apply to as broad of a spectrum of things in a catalog as possible. So, for example, one of the cases where family groupings often comes into play is with orthopedic devices, right? So in this case, you usually have devices that are made from the same material and with the same processing, but in a lot of different sizes, right? And maybe from same materials and processing, but from different suppliers and so on. So if you have something like a plate and screw set, so this is pretty common, where you have plates of different sizes and screws of different sizes, and each plate can be used with a variety of screws in different combinations and so on. You start to have to ask, okay, well, should we test every type of plate and every type of screw? So that would be like each little piece individually, or should we figure out the number of different constructs that could be made? So like 4 plates times 5 screws, 5 screw types would be 20 different constructs, or what, right? So there's I've seen folks do the construct route where, for this set of parts, they would have, 80 different possible configurations, and they, are expecting to do 80 different evaluations and full tests, right? But it really doesn't need to be that way. So if you, can carefully define a family based off of this, you can say, well, all those plates are made from the same material, same processing. It seems like the largest one that has the greatest surface area would be worst case. So we take the largest plate size from those. We can make the same argument with the screws, then we would take the largest screw size out of that. One of the questions that often comes up with parts like these is, does the color of anodization make a difference? The answer is, no, right? So titanium anodization, for example, is just a thin film of titanium dioxide on there. Chemically and from a surface morphology perspective, it's really equivalent in terms of biocompatibility. So we don't need to care about anodized colors. For plates that come from different manufacturers or parts, if there's some good evidence that can show that those two manufacturers produce an equivalent product in terms of surface residuals, things like that, then certainly you can justify that. If you can't really prove that the two manufacturers make something that's equivalent, then you can just pool those together into one family. So you can have multiple plates in there. So in the end, what could be 80 different sets of tests or 36 different sets of tests, if you go by line item, you really could represent the entire collection with just 1 or 2 sets, depending on how it's done. When it comes to actually executing this, so I said, first you do this biological evaluation plan, you make this family grouping. Another part of that is understanding what biological endpoints need to be addressed. So this is where, again, referring back to that table A1 in 10993, where that comes into play. So you can see for an continuing on with the orthopedic implant example, this would have permanent contact with tissue bone. In the document there, first you need to know physical and or chemical information. So that's part of this. We knew what materials made from and so on. And then you have E's for all of these different endpoints that need to be addressed one way or another. Cytotox, sensitization, irritation, MMP, acute systemic, subacute, subchronic, chronic, implantation, and so on. So then the question is, if we don't have to do all of those as a test, how are we going to evaluate those? And it really matters how much good information that you have to start with. So for example, if you have no existing information other than that they're a titanium part, for example, then this is really kind of like the worst case scenario or the most burdensome scenario that you should have to do. So you'd for cytotox, sensitization, and irritation and MMP, the FDA has made it very clear that they're not going to take chemistry data to address those points. So it either has to be history of clinical use or more in-depth knowledge of the material to address those. So you would have to do the biological testing for those. For all the others, except for implant, you can do chemistry testing and some sort of toxicological risk assessment. And then finally, implantation involves, really like a local interaction of the part with tissue where it touches the patient. And so that would need to be a biological test. So If you had no useful information, that's what it would look like. If, on the other hand, if you have lots of useful information, really the only tests that you might have to do are cytotoxicity and MMP, material-mediated pyrogenicity. Everything else can be addressed with a written assessment and chemistry data, depending on what you have. So, for example, for the plates that I just showed, if they have a really good cleaning validation on that, they have good cleaning validation data. We've done it several times in the past where we can say, okay, look, this is a really well-known material, CPTI. It's passivated to some sort of official spec. This has been tested over and over and over again. So there's nothing intrinsic about titanium and this passivated surface that could be a concern. And then the cleaning validation proves that in addition to the material, being known to be safe, there's nothing on it that could be of toxicological concern. So we say it's a known thing made to some spec that's very well defined and it's clean. Therefore, we don't really have a concern from the point of view of biocompatibility. And then all of this is just written in a very nicely organized way. Okay. If you do need to do chemistry testing and an assessment, then this chemistry evaluation and the details of how you do that is a whole big wide world that I love really a lot. And there's one part of this whole process that's really, really important. And that's understanding how sensitive this chemical analysis needs to be in order for it to be acceptable for a toxicologist and the FDA. And, while that didn't sound very exciting as it came out of my mouth just now, I mean, I'm trembling with excitement because it is really actually very, very important. This thing, how sensitive do you need to be when you do chemistry on a medical device, has been hotly debated for years. And, I've had lots of hot debates myself on this very topic. And the reason is that when you run a chemistry test and you get something like a chromatogram, you'll see a whole bunch of peaks, all with different magnitudes. And it's, from a practical perspective, impossible to analyze them all, right? So you need to analyze most of them, but it can be kind of difficult to decide, okay, which peaks or which compounds am I not going to identify and analyze, because you just know you have to stop somewhere. And it can be kind of confusing to know how far in this analysis is enough, right? And that's what this ISO standard addresses. So this is ISO 21726. It was, really driven forward. And, the main driver behind this is a man named Alan Hood. So he's a toxicologist with the FDA that we worked with on it. And so the goal here is to minimize the risk associated with these compounds that we leave behind, that we don't report because their concentrations are so low, accepting that we can't analyze for everything. And from a practical perspective, this can have quite a large impact on the cost of doing a chemistry test as well, if you're going to do this for biocompatibility. So the more sensitive your analysis is, then the more time it takes some expert analyst or chemist to go through the data, and the cost scales up dramatically. So it's really good to know how sensitive do you need to be from a patient safety and from a practical perspective. Okay, so this idea of how sensitive do we need to be, it borrows from this ICHM-7 concept of the threshold of toxicological concern. And so the threshold of toxicological concern is an idea that if there's a compound, even if we don't know what it is, we can feel pretty sure that it's not going to be a risk if it's below a certain threshold. So how this came about was, toxicologists have known about this issue for a long time. They said, okay, well, what's a concentration of a compound so low that if it's that low, we don't care about it? And they said, well, let's think about the nastiest compound that we can imagine. And the nastiest compound they can imagine is something like a positive control for carcinogeneticists in toxicity experiments. They said, okay, well, we know at what concentration that causes cancer every time. We're going to take something like 1,000-fold lower than that, and we'll say, okay, if it's that low, then we feel pretty good that it's not going to be a concern. And that's where these numbers come from, right? So depending on how long the exposure is to a person, the value changes. So for something like an orthopedic implant device, the TTC is 1.5 micrograms per day. So that means 1.5 micrograms of anything for the rest of that person's life wouldn't be, concerning. For shorter term contact devices, then that value goes up. So if you have a limited contact device, it gets to be a lot more convenient. This value of 120 micrograms per day is actually pretty high. That means if you do chemistry testing, it's unlikely that there's very many peaks that are that high, and you can save a lot of time that way. So if a lot of devices that we look at fall into either of these categories when we do chemistry testing. So there's lots of devices that contact people for less than 30 days. And there we would use 120 micrograms per device or per day. And then for longer term devices, it often is coming down to this 10, depending on how long the device is used, of course. Now, when you're considering this threshold, and the document explains this, you really need to also understand the uncertainty in the chemistry measurements that you're making as well. So if the number reported from a chemistry lab is only good to like plus or minus a factor of 2, then you would need to account for that in this threshold. So I think I kind of like summarized this point, but I'll just, reemphasize it here. So the point of ISO 21726 is to provide a baseline number that you can compare chemistry results to, either comparing to the maximum concentration of an identified or an unidentified compound from a medical device. If you want to support toxicological equivalence of a device, so if you have your device and a predicate and you set them side by side and they both have everything below the TTC, then you can claim that they're equivalent. Or if you compare, it's good to compare to a maximum dose exposure of any identified constituent or compound. And again, this goes directly back to what we call this analytical evaluation threshold. The TTC sets the vertical position of that line there that you see. You can see all of these noisy peaks. Anything that has a peak height above that peak is going to require all this work to identify and quantify. And you want that line, the analytical evaluation threshold line, to be low enough to be protective of patient safety, but not too low so that you're spending $100,000 trying to identify peaks and things like that. Okay, so with that, that's actually, I think I finished a little bit quicker than what I expected, but there's plenty to talk about. We're going to go in depth on the MDRs at 2 o'clock with Bo Rollins from Convatec. And we have plenty of time now to answer any questions either together, or I'm happy to answer them one-on-one with you.